Good evening, everyone. This is another opportunity for us to meet at Omega Bio-Edutainment. On your screen tonight, we are going to consider introduction to do it yourself in bioinformatics. And today we are going to look at the essential digital lab bench of bioinformatics. So don't forget that you are requested to do the task. Then thereafter, you have to submit at the email given before, and you have to go submit the hard copy later on Monday. Don't forget that, just like I said, that only those that are able to submit at the Gmail today, those are the ones that are expected to also submit on Monday, the hard copy. Now, let us just run through the content of what you are going to do. It is going to be divided into three parts, and we are going to focus on how can you use your smartphone or your laptop, how can you use it to do an offline microbial identification. Thereafter, we will look at what are the do-it-yourself techniques that we can use for protein searching and protein generation. Then there's going to be a step-by-step of how can we annotate certain proteins so that you're able to know the functions of those protein. Don't forget that, just like I said, each slide is going to be displayed for you for certain number of minutes, and you are expected to take notes and do the task as we go on. This is going to be the first DIY step, and that is displayed for you on what you have on the screen. So I want you to do step 1 to 4, then thereafter we'll go to the next slide that is going to be the conclusion of the first task. The task is actually divided, just like I said, it's going to be divided into three. So I want you to quickly rush to start working by using the workflow and steps as it is being displayed. And the first one is going to be the power of blast. So you are expected to do it, and we will see you in the next slide. So don't forget that this is going to be on for like 5 to 10 minutes, between 5 and 10 minutes. So you are expected to do that. Yeah, yeah. Once again, I want to really welcome you once again. This is another opportunity and to make a Bio-Edutainment for a do it yourself approach. Don't forget, it is like this is our own digital laboratory, and we are considering essential bioinformatics databases. In this work, DIY, three areas are going to be in focus, and that's going to be on microbial identification, protein searching, functional proteins, as well as annotation. The first workflow we are going to consider is a blast, which we are very much familiar with right away from our introduction to bioinformatics. So the step one is to understand sequence alignment. Why do we blast? Don't forget that just to compare uh, a specific protein that you are a specific protein or specific sequences that you have in hand with the set of sequences that are similar to it, even in the gene bank. So that is the workflow. Step one, understand sequence alignment. Step two, assess NCBI blast. Step three is select nucleotide blast. You know all this. Step four is to enter our query sequence. For example, you can paste your 16SR ribosomal RNA or any nucleotide sequence that you want. So that's going to be what you will do. You will enter in the space that you have to black. That is what is called query. Don't forget the difference between a query sequence as well as a subject sequence. Okay. Step five now, it is going to be to choose the search set. Then you have to initiate search, that is by clicking your blast. Then you should be able to interpret your results by looking at high level of similarities percentage, and the lower the e values, the more similar they are, which indicates strong matches to known sequences. So, and if there's any problem you want to troubleshoot, so you have to be aware of potential issues like contaminant sequences. That is, if you are not getting very good blast results, that means you need to do a data cleaning of your sequences, and you know how you can clean your data. So it could also be as a result of a low quality data as during the time of sequencing, and it could also be as well as due to chimeras, which may also affect the results. Okay. The next one is our workflow for our interest, protein of interest. Okay, so the first one is to locate the search bar by going to Uniprot's homepage, find the search bar, typically which is labeled, then in the center of the page. Then we have to select the database, and by default, you are going to see Uniprot KB. So you are going to use the drop down menu that you are going to find on the left of the search bar to select which database you actually want to use. And you should know that most functional analysis, you have your Uniprot KB, which is the default, and there are recommended choice that. So the next one is you are going to enter your query sequence, your query. Type the name of your protein of interest. For example, it could be human ACE or a specific protein name like hemolysin E into the search bar. So the next thing is to initiate the search by clicking the search button. You review the results, that is the search result. The result is going to start loading in order to offer you potential options to be able to display them as in form of card or in form of a table. And the last step, which is step six, is to confirm whether the protein, whether what you have done is of correct entry, that is in form of card field. In the card view, you check whether your card view has a title. So, and Uniprot's entry names are typically styled as protein, the name of the protein underscore organism. For example, if you are looking for ACE, that means you are going to have ACE underscore human. If your own protein is found in Plasmodium, it's going to be where you have Plasmodium is going to be human. Then you have to confirm whether you have the correct entry by checking the protein name and the organism in the description below. So, and what you are going to do that you should do is to practice searching for a different protein, not only for this one. You can also make use of a bacterial protein like hemolysin E. So you can do that. Then, how can you functionalize your protein? How can you determine the function, annotate the protein? How can you guess? I mean, through your do it yourself, how can you guess that? So this is by focusing, you navigate to the function section still under the uniprot within the protein entry in order to be able to locate where you are going to see the function. So just click on the function, you observe the feature, you will see a diagram which is going to be often a gray bar with color shape representing the amino acid sequences. The color shapes identify section important for the protein function known as functional features. So the step 3 is to examine the feature tables. So there is going to be a table that will be displayed to you. So below the diagram, there's going to be a table listing these functional features, which is expandable in order to be able to view the entire list. So what you do here is to take a screenshot of all these so that you'll be able to submit that as well. Okay. So the next, which is step 4, is to identify features types of protein, like the common functional features, which might be it could be enzymatic active sites, that is where enzymatic reaction occur. On that bar that you are going to see, you are still going to have a section that is going to be binding sites where other molecules will be able to bind, cleavage site where the protein might be cut, then the domain that's going to be made of distinct functional or structural units within that protein. So then step 5 is to be able to filter your feature. You use the type, which is the drop down menu, if it is available in your browser, or filter for a particular feature such as enzymatic activities, I mean active site. So in order to be able to display, so if you want to know all the amino acids that are functioning or that have enzymatic active sites, it is going to display for you as a kind of filter result. So then the action for your protein, be able to identify at least one functional feature and note its type and position as well. Then if you want to navigate, if you want to know, want to enter a protein entry, once you have found your protein, the slide shows you how to understand its basic information. Access the protein entry by what? By entering the accession number, the protein number of that. For example, if you don't know the name of that protein, but you are so sure of knowing the protein number, unique number, you can also use it like that. That is, you assume that instead of you to know the protein name, what you know is just the protein number, specific number. So, and you will enter that just like what you have there to be able to access its detailed page. So the second one is to locate the summary header at the top, very top of the protein entry. You'll find the summary header uh, at which, I mean, by which you will be able to interpret your basic information. And lastly, we are going to step 4, which is going to be understand annotation score. How can you understand the annotation score? Below the description, note the annotation score. So this indicates the level of detail in the entry and how much experimental evidence supports this information. So what that is telling you is that if you have a score of 5/5, that means it's the most reliable data. If you have 4/5, is also reliable, but it's more 5 over 5 is more reliable. Then say 5, you can review the tags by looking at the tag that provide quick information. So, for example, now you might not actually need to carry out any other thing apart from what you see. You're going to see hashtag. The next thing you see is the function. So, if that protein is serving as a for toxin, you will see hashtag toxin because it is a protein that that organism is using for production of toxin, then slash virulence, that is telling you that. So for your choosing protein, you should be able to identify the name, the length, that is the number of amino acid, and the annotation scores. So having done that, I want to really wish you best of luck. Don't forget